Hello everyone, I welcome you again in my channel. So today we will be discussing about bacterial cell envelope and its components, right? So if we have to look at a broader end, cell envelope will consist of your three major parts. First one we have is the capsule, which is the outermost, that is generally for the protection of the bacterial cell. Secondly we have is the cell wall, and the third component we have is the plasma membrane. So these are the three components that we are going to discuss mainly. And if we have to talk about the contents of the bacterial cell which are present inside these three components, we will just have a pictorial representation of that, and the function of each that constituent will be discussed in the next topic. Okay, so let's start. So first let's talk about what is the major difference that you can see between your archaea and your bacteria, right? So archaea, they are known as basically the primitive type of bacteria, and the bacteria, they are also known as the eubacteria, right? But the major common point that you can consider in these both are, that is the cell type, that is they both having the prokaryotic type of cell. That means their nuclear content that is not bound by a membrane. That is the common factor in both the types of bacteria, right? But the different distinguishing points among these two are that in archaea, basically their cell wall, it is composed of a compound which is known as pseudomurine. However, in bacteria, they are composed of a compound which is known as murin. Now here you can see it is written peptidoglycan. So peptidoglycan is also known as murin, and which is a major constituent of cell wall in case of bacteria, and it is not present in case of archaea, right? So that is the first difference. Secondly is that the archaebacteria, they generally survive in the harsh environmental conditions. That's why they are known as halophiles, thermophiles, and your methanogens. However, bacteria, they can be having the symbiotic relationships, and they can survive in the normal climatic or environmental conditions that we have. Right. Then another point of differentiation is that in case of archaea, their membrane lipids are ether linked. However, in bacteria, their membrane lipids are aster linked. Means the linkages, they are varying in case of archaea as well as in case of your bacteria. Right. So if you have to talk about archaea, so here we have given a description, that is they will not have any kind of peptidoglycan in layer, and the antibiotics, they mostly act over the peptidoglycan. So since they do not have the peptidoglycan, obviously they will have no antibiotic sensitivity. However, in case of bacteria, since they have the peptidoglycan, so they will be sensitive to your antibiotics. Okay, next we have here, you can see this is a typical structure of a bacterial cell, and this is the diagram that you can see. These are the highlighted portions will represent the different components. As you can see, this is the flagellum, right, that is associated with your cytoplasmic membrane. Outermost we will have the capsule, then we will have the cell wall, then we will have the cell membrane, right? Now all the constituents inside the cell membrane, that is this portion and all this your genetic material and everything, that is known as protoplast, right? So now this protoplast will consist of your sulfur granules, which are nothing but which are the storage granules that are there for the storing of sulfur. And then next we have is the gas vesicles. Gas vesicles, they are also there, but they generally provide basically for the maintaining the cell buoyancy. Then we have the another component, that is polybeta hydroxybutyrate, which is nothing but which is a carbon storage reservoir. And finally we have is the magnetosomes, which are generally helps in the cell movement, right? These round structures, refer to them as ribosomes, and they have a primary role in protein synthesis, right? This crumbled structure is known as the genetic material, that is the nuclear material, and since it is not bound by any kind of organelle, that's why it is known as a prokaryotic cell. Okay, so this is how you can draw a bacterial cell, and these are the different components of this. And these small hair like fragments, as you can see here, they are the pili, and they have a major role in conjugation, which is a type of a reproduction mechanism, or a combination recombination mechanism, as in the case of bacteria. Okay, now next you can see, now we are focusing towards bacteria only, which are also known as the true bacteria, right? Because the bacteria, based upon their staining reactions. That means they are treated with certain kind of stain, and based upon the stain that they will acquire, when we visualize them under a microscope, they are categorized as gram negative and gram positive. Gram negative if they take the safranin, which looks like your pinkish, and your gram positive means when they take up the crystal violet stain, which looks like this, right? Now these gram negative and the gram positive bacteria, they will acquire a particular stain because of the differences in their cell wall, that we will discuss in the coming slide. But because of that differences, they are getting the particular color. Okay, now this phenomena of gram staining, it was given by the Christian Gram in the year 1884, and where he has basically distinguished the bacteria based upon their cell wall, and because of that differences in the cell wall, they will acquire the different color during the staining reaction. Okay, now here you can see what is the major difference between a gram positive and gram negative. So in gram positive bacteria, their cell wall, that is 70 to 80%, it is composed of a compound that is known as peptidoglycan, which is also known as murine, and below that cell wall the another compound is there, which is known as plasma membrane, that basically consists of your phospholipids, right? However, in gram negative bacteria, this peptidoglycan layer is very much thin, and it generally lies between two layers, that is known as plasma membrane and the outer membrane, right? So this is the reason why the gram positive will stain the primary stain, that is your crystal violet, because it has a thicker peptidoglycan layer and it get trapped inside this layer. However, in case of gram negative bacteria, this layer it get dissolved because of the action of ethyl alcohol that we added during the gram staining reaction, and it will take up the counter stain, because the primary stain get washed off, and that's why it looks like pinkish, and that is the gram negative, and it's look like purplish because of their gram positive reaction, right? Now we will study what is this peptidoglycan layer and how it is being formed, and what is the major role and component of it, right? So here you can see this is the major structure of a peptidoglycan layer. Now that peptidoglycan layer, it is also known as your murine, right? Now that murine, it will composed of basically two components, that is two glucose units, that is NAM and NAG. So NAM stands for N-acetyl glucosamine, and NAG stands for N-acetyl glutamic acid, right? Now these two units, right? So here we have the two units, that was N-acetylmuramic acid and N-acetyl glucosamine, which are your two sugar backbones, right? So these are the two alternating units of these NAM and NAG. So we have the long chains of these NAM NAG, and these long chain of NAM NAG will have a this longer chain, which is known as the tetrapeptide chains, and two tetrapeptide chains, they are bound to each other by a peptide bridge. That means, let us say we will have a NAM here, so it is having a long chain which is known as a tetrapeptide chain, and the two NAM units which are having the two tetrapeptide chains, they are bound to each other by a pentapeptide interbridge, right? Now this tetrapeptide chain will consist of your 4 amino acids, and this peptide interbridge also consists of your 4 different amino acids that we will discuss in the next slide, right? So here you can see these are your NAM-NAG units, as you can see. So this is the tetrapeptide chain that was attached to the NAM unit. Here you can see the tetrapeptide chain will include L-alanine, D-glutamine, D-aminopimelic acid, and D-alanine. Similarly, the second chain NAM will consist of the similar tetrapeptide chain, that is L-alanine, D-glutamine, D-aminopimelic acid, and D-alanine. So there is a bond linkages that occurs between the, when it is called as the beta 2,4 linkage, because it was the this one beta 2,4 linkage and beta 3,4 linkage, sorry. Okay, so in this case you can see this is a beta 3,4 linkage because it is attached with the DAP that is present at the third point. Here it is the beta 2,4 linkage because it is attached to it at the second point, right? So this is your beta 2,4 linkage, and this is an example of your beta 3,4 linkage. ठीक है. Now this beta 2,4 linkage and beta 3,4 linkage is there. They are both attached to each other by a peptide interbridge, which will consist of your 5 units of particular amino acid. Like here, these 5 units are of glycine, right? So that is your peptide cross linkages, which was there in your gram positive and gram negative bacteria. Generally, the gram negative bacteria will have the beta 2,4 linkages, and gram positive bacteria will have the beta 3,4 linkages, and the gram positive bacteria will have a higher concentration of that peptidoglycan. However, the gram negative bacteria, they have a lower concentration of that peptidoglycan. Okay, so that is the major difference. Now this we have discussed in case of your gram positive bacteria, that they will have a higher content of peptidoglycan layer, right? And their 2 units are there only. One is the peptidoglycan, and below that we have is the cell membrane. However, in case of your gram negative bacterial cell, 3 units are there. One is the common one, that is your cell membrane, and the in between we have is the peptidoglycan, and the one that is above the peptidoglycan layer is known as the outer membrane. So basically you can see here that the peptidoglycan, it is interlinked between your outer membrane and your cell membrane, and it is kind of a small or minor, or you can say smaller in quantity as compared to your gram positive, right? Here you can see this is the normal cell membrane having the phospholipids. However, this is the this your peptidoglycan layer, which is present in a region that is known as periplasmic space, right? So this is a question that normally arises in many of the competitive exams, be it net, be it any kind of ADO and some other exams related to agriculture. So here common question is that the periplasmic space is present in which type of bacteria. So a common answer to this will be a gram negative bacteria, right? So these are the two regions. Third and a new type of region in case of your gram negative cell wall is the outer membrane. Now this outer membrane will consist of a important compound which is known as lipopolysaccharide. Now what is the main role of this lipopolysaccharide in this gram negative bacterial cell? It is that basically if we compare the gram positive and gram negative bacteria, most of the plant diseases, they are caused by your gram negative bacteria, right? So basically this gram negative bacteria, they have this lipopolysaccharide which will have the antigenic properties. Antigenic properties means that they will, whenever a particular bacteria will infect a plant, they will release certain antigens that was being recognized by the plant antibodies, and that's why a kind of a recognizing relationship will occur, and that recognizing relationship is carried out by that antigenic properties, and which was given by your lipopolysaccharide, right? So that is a complete structure of a gram negative cell wall, right? Now next you can see whatever we have discussed, this is basically given here in tabular form. First, in gram positive, obviously high concentration of peptidoglycan, so it should be thick. Secondly, peptidoglycan layer is present in abundance. Thirdly, since the peptidoglycan layer is abundance, obviously the lipid content is less. And here the most important component, that in gram positive cell wall there is a teichoic acid which was absent in gram negative cell wall. Similarly, in gram negative cell wall there was the lipopolysaccharide which was present, which was absent in gram positive cell wall. So the role of lipopolysaccharide is that it will have the antigenic properties. Now what is the role of this teichoic acid? The teichoic acid basically helps in providing the flexibility to the cell wall, right? So the flexibility to cell wall is being provided by the gram positive cell wall, and it also has a role in conjugation, right? So it has a also role in conjugation, right? And the last point that will differentiate between a gram positive and a gram negative cell wall is that in gram positive lysozyme is there that will form the protoplast. Now lysozyme is what? Lysozyme is basically an enzyme that will act on the bacterial cell wall and break its linkages, particularly your beta 1,4 linkages, right? So now since the gram positive cell wall will have only the peptidoglycan layer, so it will act over it and it will consume all of its content, and whatever we are left with is the protoplast, right? So we can develop the protoplast from the action of lysozyme on the gram positive cell, right? However, in case of your gram negative cell wall, since the peptidoglycan, it is a middle layer that is being sandwiched between the outer membrane and the inner membrane. So if we consume that, we will have some portion of your periplasmic space and your inner membrane left, right? So that is why they are known as the spheroplast, where the remnants of the cell wall are present even after the application of your enzyme, that is lysozyme, right? So that is a kind of a major differences between the gram positive as well as your gram negative cell wall. Okay, I hope everything is clear. If you want to have any clarity or if you have any kind of doubts, you can comment down in the comment section. Now next we have the most important part. Now this, you can see, this is the structure for bacterial flagella, right? Now bacterial flagella, as we have already discussed, that they have a role in bacterial movement. They are present in both gram positive as well as the gram negative. There are three components of any bacterial flagella. First one is the filament, that is composed of a protein flagellin. Second one is a hook that basically attaches your basal body with the filament, and basal body will have different kinds of rings that are being embedded in the cell wall and the plasma membrane, right? So if we have to talk about gram positive cells, so since gram positive cell have a high content of peptidoglycan layer, so they already have that kind of firmness, right? So that's why only two types of rings are present in bacterial flagella in the gram positive cell, that is known as the L ring and the P ring, and they are being attached at the outer membrane, right? However, in case of the gram negative bacteria, since the peptidoglycan layer is lies in between your outer and inner membrane, so it is somewhat fragile, so that is why it requires more kind of strength to hold it together. So that is why in case of gram negative bacteria, we will have all the four rings present, that is your L and P rings, which are attached in the cytoplasmic membrane, and your MS and C ring, which are attached into their inside of your cytoplasmic membrane, right? So this you have to always remember. There are four rings, L, P, S and C, right? So among this LP ring, they are present only in the gram positive, but your LP, MS and C ring, they are present, that is four types of rings that are present in gram negative your bacteria, right? This is also very important questions where it is being asked that these are the different rings and in which bacteria they are present. So if the four rings are there, obviously they are present in any gram negative bacterial cell, and if only the two rings are there, they are present in only the gram positive bacterial cell. Okay, so now another component which we have not discussed, and we will discuss about it in more depth in the later slides or in coming lectures, that is the plasmid. Now here you can see this is the bacterial DNA, this is the plasmid. So plasmid, how you can define basically a plasmid, that plasmids can be defined as the small circular piece of DNA. This is the entangled form of DNA. These are circular forms of DNA, but they are independent of your bacterial DNA, and they have the ability to replicate independently, and they also have the chromosome. Now why there is a need that the bacteria will have a another independent chromosome if already they will have the nuclear membrane? That is because of the fact that these plasmids or the circular nuclear DNA, they have a role to play in conjugation, etc., and in some cases they also provide the kind of a resistance that is being provided by these kind of bacteria to certain against certain pathogens or against certain harsh environmental conditions, right? For example, we have different kinds of plasmids like conjugative plasmids, non-conjugative plasmid, F plasmid, col plasmid, bacteriocins, colicins, etc., right? So these are the different plasmids based upon their different activities or based upon their different functions, right? So this we will discuss in the more coming lectures. So now you can comment down to if you want to have any other special lecture. Otherwise, for next topic, I think we should discuss about the wheat, which is a very important crop. So we'll discuss about the wheat, particularly the three important diseases of wheat, that is the stem rust, that is your rust, basically having the stem rust, brown rust and your yellow rust, then your bunts, and then your smuts. Or we can also discuss some other topic if you have any. So kindly go through it, and if you have any queries you can comment down. So thank you all for watching this, and I'll require a lot of support from you guys so that I can teach you as long as possible, because this is my passion. I want you to please support me. So like, subscribe and share, and please comment down if you have any kind of special request, or if you want me to make any kind of special series particularly for the plant pathology students. So thank you very much and have a great day.